All right, ladies and gents, welcome back. So today we're going to finish our discussion on cell transport and really dive into the actual types of active transport that your cells can perform. So remember that the big difference between active and passive transport is the amount of energy that your cells expand, plus the direction in which those molecules are moving. So with passive transport, I don't need energy and I move with the concentration gradient. So I'm going from an area where those molecules are already high, moving them down the concentration gradient to areas where they're low. In active transport, I'm now going to go the opposite direction. So now I'm going to move these molecules against their concentration gradients. So now I'm moving them from an area where they're low to an area where they're already highly concentrated. So I'm forcing more molecules into an already crowded space. And as a result of that, I'm going to need my cells to push those molecules in that direction. And so I'm going to need energy in order to do this. Now, a lot of active transport processes work as these pump systems, like you see here. And so these pump systems work really in four basic steps. First step is just the solute bonding to the protein, the transport protein that I'm going to use. I'm going to break down that ATP, make ADP as a result, get that energy out, take that phosphate, phosphorylate the protein. When I do that, it causes that protein to change its shape, both in terms of its orientation to which side of the membrane it's open to, as well as if you look, the bonding site to those solutes also changes. That changing of that bonding site forces the solute back out, and once that solute is released, that causes the protein to change shape again and force the phosphate back off. So it's solute bonding, phosphorylating the protein, actually changing the protein shape, causing the transport to actually occur, and then the protein will reverse its configuration, reverse its shape, dephosphorylate, and then the cycle can start over. Now, one of the biggest, most important active transport pumps in your cells is your sodium potassium pump. And this one is incredibly important in things like your nerve cells, because what this is going to do is allow me to set up charges that are different inside my cell membrane versus outside. So I set up what we talk about as an electrical impulse, or electrical differential. And as your cells send those electrical signals down those nerve cells, that charge flips. So it might have been more positive here, more negative outside. And as the signal goes, it now flips that charge. But your cells need to be able to reset that charge differential again, and that's where your sodium potassium pump comes in. So your sodium potassium pump allows your neurons to basically reset so that they can send another signal. And so your sodium potassium pump works in really 6 big steps, 3 that have to do with the sodium ions and 3 that have to do with the potassium. So if I take a look here, all right, I'm going to start where I've got really high sodium outside, not a whole lot of sodium inside, a lot of potassium outside, but not a lot of potassium inside. And so remember that this is active transport. I need ATP because I'm moving against the gradient. So I'm going to start here where I'm going to bind 3 sodium ions from inside my cells where the sodium is already low. All right, we're going to break down the ATP. We're going to phosphorylate the sodium potassium pump protein. When I do that, it causes the pump to change its shape. The bonding sites for those sodiums change, and it forces those sodiums outside of the cells. Remember, it's already high out here. So I'm now forcing even more sodium out into the external environment of my cells where it's already high. Now, while that phosphate is still bound and that pump is still open, I'm going to load 2 potassium ions. Again, low potassium outside my cells. That loading or that binding of that potassium causes the protein again to change shape. That kicks off the phosphate, opens that protein back to the inside of my cells. All right. And in that process, also changes the bonding sites for the potassium. Potassium unloads. It's now inside my cells where it once again is already high. Okay, and then the whole process starts over again. Load the sodiums, phosphorylate the protein, open it to the outside, release the sodium, load the potassium, dephosphorylate the protein, and then unload the potassium, and the cycle continues. Now, the sodium-potassium pump and other pumps like it have to do with moving molecules against their concentration gradients, and as a result, we need the protein pump and we need the ATP. These last two types of active transport really don't have much to do with the concentration gradients, but instead go back to that size idea. Remember that if I'm going to move materials across that cell membrane, they need to be relatively small. But there are situations where your cells produce large molecules, big proteins like hormones or waste products or enzymes, and I need to get those materials outside of my cells, especially if I'm talking about a multicellular organism like us. So how are they going to get these big molecules out if I can't get them just squeezing them through a protein in the membrane or squeezing them through the phospholipids? And that's what this process of exocytosis comes in. So let's break this down. If I look at this, exo is like exit, and I'm exiting the cell. Cyto is cell. So I'm going to release these large molecules from the cells. Go back to that idea of the endomembrane system, right? We talked about your nucleus having the set of instructions, your rough ER with the ribosomes attached built the proteins. We load them into a vesicle. That vesicle goes to the Golgi, where the Golgi decides what to do with it, finish the protein, pack it up into another vesicle, and if it decides it needs to be exported or secreted or excreted, depending on what term you come across, then that vesicle, here's the vesicle from the Golgi body, will come to your cell membrane, merge with that cell membrane, and then spew those contents out. And so these may be hormones. Hormones are often proteins, and something like your thyroid gland, for example, makes a couple of different proteins, a couple of different hormones. Those hormones get released into your bloodstream, and then they'll go to other cells like your stomach and your liver, and they help with digestion and help with hunger pains and things like that. But in order to get them out of that thyroid has to be able to move these big molecules out, and so they'll do it through exocytosis. Now, what if your cells need to come in the other direction? Then this is where we talk about endocytosis. So exo like exit, endo is within. So now I'm going to bring these big molecules in. And a lot of times these are food particles. So that membrane folds in, creates a vesicle, and here are those food vacuoles, right? And those food vacuoles will go then merge with a lysosome, which contains your digestive enzyme, break those food particles down, and be able to use those molecules then for other metabolic processes. Now, the big difference here and why endo and exocytosis are both considered active transport is because I need ATP to make and move these vesicles. So the ATP energy expense isn't so much about moving things against their concentration gradients, but now it's about moving these molecules around. So when we come back here, this is the transport work that we talked about, right? Go back here. So here's where the sodium potassium pump and things like that come in. When we get to endo and exocytosis, now we're really talking more this mechanical work. I need that phosphate to move these motor proteins around, and your vesicle is going to sit on the back end of that motor protein so that I can move them either by creating them and moving them to the cell membrane, like I'm doing here in exocytosis, or creating that vesicle and then moving it somewhere inside my cell via endocytosis. Now, endocytosis can also have two subcategories depending on what kind of state these molecules are that I'm bringing in. If they're big solid particles, we talk about it as phagocytosis. All right, so these would be large solid particles. Something like your white blood cells. How do they actually help you fight off an infection? Well, some of them are phagocytotic cells. These would actually be little bacterial cells that your white blood cells create a vesicle around, bring it in, merge it with a lysosome, dissolve that bacteria, and hopefully that then is keeping you from getting sick. If these are large liquid particles instead, then we'll talk about it as pinocytosis. And same idea, we'll bring those molecules in, break them down, use them for whatever purpose that we need.